Highly radioactive graphite blocks at the Chernobyl power plant support the growth of specialized black fungi. These organisms thrive in environments where ionizing radiation levels are lethal to most known life forms. Radiotrophic fungi possess a unique metabolic capability called radiosynthesis. They convert the energy from ionizing gamma radiation into chemical energy to sustain cellular growth and biological functions. The primary component enabling this energy conversion is melanin, a complex pigment found in the fungal cell walls. Melanin acts as a biological transducer, capturing high-energy photons and directing their energy into metabolic pathways. In standard biological systems, ionizing radiation causes the radiolysis of water, creating reactive oxygen species. These radicals shatter deoxyribonucleic acid and denature proteins, leading to rapid cell death. Fungal melanin provides a physical shield by absorbing gamma rays. However, it does not merely block the radiation. It utilizes the absorbed energy to change its electronic state and initiate charge transport. When a gamma photon strikes the melanin polymer, it ejects electrons or excites them to higher energy levels. This ionization event is the first step in the conversion of electromagnetic radiation to chemical potential. Melanin is composed of indole subunits linked into a disordered three-dimensional network. This structure allows for extensive pi electron delocalization, making the pigment behave like an organic semiconductor. The excited electrons from melanin are captured by electron carriers within the fungal cell. These electrons enter the mitochondrial electron transport chain, driving the synthesis of adenosine triphosphate. Under ionizing radiation, radiotrophic species exhibit a significant increase in the rate of acetate uptake. This accelerated metabolism allows the fungi to grow up to 3 times faster than non-irradiated controls. Cladosporium sphaerospermum is one of the most prominent radiotrophic species found in Chernobyl. It displays a remarkable ability to survive radiation doses exceeding 500 gray. Experiments demonstrate that these fungi exhibit radiotropism, which is growth directed toward a radiation source. They sense the radiation gradient and steer their hyphae toward areas of higher flux to maximize energy intake. While plants use visible light photons to reduce carbon dioxide, radiotrophic fungi utilize high-energy ionizing photons. Both processes represent the biological capture of electromagnetic energy to power life. The quantum efficiency of melanin in converting gamma radiation is lower than chlorophyll in sunlight. However, the sheer energy density of ionizing radiation compensates for this, providing a substantial power source. Melanin also functions as a powerful antioxidant. It neutralizes the free radicals produced by radiation before they can interact with the genome, acting as both an energy harvester and a protective shield. Radiotrophic fungi possess highly efficient deoxyribonucleic acid repair enzymes. They can rapidly mend double-strand breaks that would be catastrophic for other eukaryotic organisms. Many of these fungi are polyploid, meaning they carry multiple copies of their genome. This genetic redundancy ensures that even if one sequence is damaged, functional templates remain available for repair and replication. Cryptococcus neoformans, a human pathogen, also exhibits radiotrophic growth. When melanized, it survives massive radiation exposure by redirecting the energy to bolster its cellular defenses. Irradiation changes the oxidation-reduction potential of melanin. This shift facilitates the transfer of electrons to nicotinamide adenine dinucleotide, which is a vital cofactor in metabolic energy production. The growth of these fungi is a classic example of non-equilibrium thermodynamics. They maintain high internal order by capturing external high-quality energy from the decay of radioactive isotopes. Samples of Cladosporium were sent to the International Space Station to test their effectiveness as biological radiation shields. The fungi continued to grow and attenuate cosmic rays in the microgravity environment. For future Mars missions, radiotrophic fungi could be cultivated to create living radiation shields. A layer only a few centimeters thick could significantly reduce the radiation dose for human inhabitants. These fungi can be used for bioremediation to stabilize radioactive waste. By incorporating isotopes into their biomass, they prevent the migration of hazardous materials into the surrounding groundwater and soil. The presence of melanin increases the velocity of nicotinamide adenine dinucleotide cycling. This allows the fungi to maintain high levels of reducing power even when traditional nutrient sources are scarce. Melanin acts as an amorphous semiconductor. Its conductivity increases with hydration and temperature, allowing it to interface directly with biological electron transport chains under varying environmental conditions. Decades after the disaster, the fungal populations inside the reactor continue to evolve. They have adapted to constant high-flux environments, showing genomic stability despite the extreme conditions. Gamma-ray attenuation in melanin occurs through the photoelectric effect and Compton scattering. These interactions convert high-energy photons into kinetic energy of electrons within the pigment matrix. The biomass yield of radiotrophic fungi is directly proportional to the radiation intensity. This linear relationship suggests that radiation is the primary limiting factor for their growth in nutrient-poor environments. Hyphae can penetrate porous radioactive materials, extracting energy from the interior of contaminated structures. This deep infiltration makes them ideal for treating structural contamination in decommissioned nuclear sites. Fungi synthesize melanin from tyrosine or dihydroxyphenylalanine. The polymerization process is enzymatically controlled to ensure the resulting pigment has the necessary electronic properties for radiosynthesis. Beyond gamma radiation, fungal melanin provides exceptional protection against high-energy ultraviolet C radiation. This makes these fungi candidates for terraforming or surviving on planetary surfaces with thin atmospheres. Some radiotrophic species can actively bioaccumulate cesium-137 and strontium-90. They concentrate these isotopes within their fruiting bodies, facilitating the physical removal of radiation from an area. Research suggests that radiotrophic fungi may perform extracellular electron transfer. They could potentially donate electrons to minerals in their environment, influencing the geochemistry of radioactive waste sites. Comparative genomics reveals that radiotrophic fungi have upregulated genes for deoxyribonucleic acid repair and oxidative stress response. These genetic adaptations are essential for maintaining viability under chronic irradiation. Fungal biofilms have been observed in the cooling pools of nuclear reactors. These films survive in pure water environments by utilizing the intense Cherenkov radiation as a supplementary energy source. Cherenkov radiation is emitted when charged particles travel through a medium faster than the speed of light in that medium. Fungi capture this blue light using melanin and other light-sensitive pigments. Melanin can store electronic charge for short periods. This allows the fungi to maintain metabolic activity even during fluctuations in radiation intensity, acting as a biological capacitor. The ability to utilize radiation may have evolved early in Earth's history when the planet lacked an ozone layer. These ancient metabolic pathways are now being rediscovered in modern radioactive hotspots. Unlike most proteins and lipids, melanin is chemically inert and extremely stable. It does not degrade even after absorbing massive cumulative doses of ionizing radiation, ensuring long-term protective functionality. The proton gradient generated by melanin-mediated electron transfer drives the ATP synthase motor. This enzyme converts mechanical rotation into chemical energy, producing the fuel needed for cellular repair. Fungi sense radiation through the production of specific signaling molecules triggered by melanin excitation. This biochemical signal transduction allows the organism to navigate complex radioactive environments effectively. Engineering radiotrophic fungi could lead to self-repairing organic coatings for nuclear facilities. These living materials would strengthen over time as they consume the ambient radiation of the site. The semiconducting properties of fungal melanin are being explored for organic electronics. Melanin-based transistors could operate in high radiation environments where traditional silicon-based components would fail. While radiation increases mutation rates, Radiotrophic fungi manage this through precise deoxyribonucleic acid polymerase activity. They balance the need for adaptation with the necessity of maintaining genomic integrity. Biological shields grown from radiotrophic fungi could protect astronauts from galactic cosmic rays during multi-year voyages. Unlike lead or water, these shields are self-healing and can be grown in transit.
Radiotrophic fungi also show high tolerance to heavy metals, often found in nuclear waste. Their cell walls bind to cations like lead and uranium, preventing toxic interference with internal biochemistry. Melanin exhibits a strong thermoelectric effect, converting temperature gradients into electrical voltage. This property may allow radiotrophic fungi to harvest thermal energy from radioactive decay in addition to photon energy. Radiotrophic fungi are not limited to Chernobyl. They have been discovered in high-altitude environments and near deep-sea hydrothermal vents, highlighting the versatility of melanin-based energy systems.
While radiation provides energy, the fungi still require carbon and nitrogen sources. Radiosynthesis acts as a metabolic supplement, allowing the fungi to survive in environments where organic matter is extremely scarce. Synthetic biology aims to transfer radiotrophic genes into other organisms. This could create radiation-resistant crops or bacteria designed for environmental cleanup in the most contaminated zones on Earth. The discovery of radiotrophic fungi at Chernobyl has fundamentally changed our understanding of life's resilience. These organisms prove that even the most destructive forces of physics can be harnessed as a source of life.